I am doing prompt 2-1 with single dominant epistasis. The single dominant epistasis means that an allele at one locus sets the phenotype no matter what kind of allele is at the second locus. I am going to explain this in terms of a variety of fictional mushrooms in the mushroom kingdom. There is a species of mushroom which comes in 3 different types: the bad mushroom, the wilt shroom, and the amanita. Which of the types the mushroom will turn into is based on 2 genes. If the first enzyme is inhibited, the mushroom will be a bad mushroom. If the first enzyme is synthesized and the precursor is allowed to turn into the intermediate, then the mushroom would be a wilt shroom. If the second enzyme is synthesized and it will allow the intermediate to turn into the product, allowing the wilt shroom to become an amanita. In the first gene, the dominant allele is a repressor. This means that if there was a dominant allele with the first locus, the inhibitor would prevent the enzyme from converting the precursor to the intermediate. Since there are no intermediate, it does not matter what the second locus is, it won't be able to turn anything into the product. In this diagram, the arrows are alleles that are involved in producing enzymes, while the T-bars are alleles that are inhibiting the enzyme. The arrow with an X is the gene that won't produce the enzyme. The dominant gene is the second locus, is what causes the second enzyme to be synthesized and so will complete the transition to the product. For the purposes of single dominant epistasis, you do not need to know the identity of the second allele if the first allele of that locus is dominant, as long as you are not breeding them any further. You have probably heard of Punnett squares. This is a crossbreed with a single locus without epistasis. Here there are 2 loci, which means there have to be 2 alleles for each category in epistasis. In theory, I could make 2 separate Punnett squares, but that would make things a little more complicated to get the same result. With a crossbreed between capital A lowercase a, capital B lowercase b crossed with a capital A lowercase a, capital B lowercase b, results with 9 capital A capital B offspring, 3 capital A double lowercase b, 3 double lowercase a capital B, and 1 double lowercase a double lowercase b, which gives 12 bad mushrooms, 3 amanitas, and 1 wilt shroom. All the ones colored red are bad mushrooms, yellow are wilt shrooms, and green are amanitas. With a crossbreed between a capital A lowercase a double lowercase b and a double lowercase a capital B lowercase b, you get 4 capital A capital B offspring, 4 capital A double lowercase b, 4 capital lowercase a capital B, and 4 double lowercase a double lowercase b offspring, which gives 8 bad mushrooms, 4 amanitas, and 4 wilt shrooms. And here are my citations.